Hello everybody. I'd like to start off by thanking you for giving me the opportunity to present my research today, which I hope you'll find very interesting. The title of today's presentation is Imaging Cancer Stem Cell Antigen CD133 with a novel Zirconium-89 labeled antibody. Before I get into details, I would like to introduce to you McMaster's Radiochemistry group, who I have been doing my PhD with for the last 3 years. Our team specializes in non-invasive imaging and therapeutic modalities for imaging and treatment of oncology, inflammation, and infection. Our approach is holistic. We select and synthesize small molecules and proteins, then develop these into probes. We test the new probes using a variety of in vitro and in vivo techniques. Lead imaging probes and therapeutics are adopted by our clinical and commercial partners, including the CPDC, Fusion Pharmaceuticals, and companies founded by graduating students. Traditional screening methods for breast cancer rely on mammography, where suspicious lesions are followed up by 18F-FDG. While mammography is limited in its capacity for detecting breast cancer, especially in its early stages, 18F-FDG offers little towards understanding the biology and prognosis of breast cancer patients. That's why we pursued novel PET agents for imaging breast cancer biology. CD133 represents an attractive biological target for imaging breast cancer and for guiding patient treatment. CD133 identifies cancer stem cells, a treatment-resistant tumor subpopulation liable to repopulate tumors following therapy. The antigen also predicts breast cancer patient survival. The figure I'm showing you comes from a study where archival breast tumor biopsy samples were characterized for various biomarkers, including CD133, and compared to patient outcome. The blue line on the figure represents patients whose tumors were CD133 negative. This cohort of patients had the highest survival probability. Patients whose tumors express CD133 on their cell membranes represent the cohort with the lowest probability of survival. While not shown, we also found CD133 represents breast cancers of the most treatment-resilient kinds, including triple-negative breast cancer. The next generation of imaging probes for breast cancer will rely on identifying important cancer biomarkers like CD133 for personalized breast cancer patient care. While valuable for its presence in treatment of resilient breast cancer, cancer stem cells represent a challenging tumor subpopulation for imaging because of their rarity. In this slide, we characterize CD133 expression in a model tumor cell line, the HT29 line, used throughout this presentation for studying CD133 in our in vitro and in vivo studies. The CD133 positive cancer stem cell population in this cell line was less than 10%, and immunohistochemical analysis demonstrated heterogeneous antigen expression, tending to localize in the tumor core. Cancer stem cell rarity thus presents an immediate obstacle for PET imaging probes that we were required to overcome. Our strategy for imaging the rare CD133 positive cancer stem cell population is driven by a novel fully human anti-CD133 antibody developed by our collaborators at the University of Toronto. The antibody was developed by phage display for binding to CD133 with excellent sub-nanomolar affinity. Its fully human structure offers minimal immunogenicity in patients, improving performance and easing translation into the clinic. In our labs, this antibody is functionalized for PET imaging using a robust two-step radiolabeling strategy. Then the antibody can deliver positron-emitting radioactive isotope zirconium-89 to target cells. Zirconium-89 is an ideal radioisotope for PET imaging using antibody-based delivery, owing to its half-life of 3.3 days. This pairs well with antibody pharmacokinetics, which typically show maximal tumor uptake after several days post-injection. After synthesis, our team evaluates the radiolabeled probe for binding affinity, immunoreactive fraction, and plasma stability before testing its tumor uptake and specificity in tumor-bearing mouse models. This is where my experimental work began. I attached 3 to 4 chelators to the antibody, chelators designed to tightly and stably bind zirconium-89, using a biologically mild conjugation reaction. Using MALDI mass spectrometry, we showed an increased molecular weight for the antibody conjugate relative to the unconjugated antibody, which corresponds to 3 to 4 chelators per antibody. By flow cytometry, we found no differences in the EC50 for the antibody conjugate relative to the unconjugated antibody, showing that binding affinity was preserved through the conjugation reaction. Next, I radiolabel the antibody with zirconium-89 using well-established procedures. The radiolabeling reaction is fast and high-yielding, resulting in a highly pure probe with great radiochemical characteristics that I show in the bottom left-hand corner. The radiochemistry and characterization used in this reaction is in line with GMP testing and combined with the clinical advantages offered using a fully human antibody, we consider this probe as specifically designed for clinical translation. Before in vivo testing, we confirmed the high binding affinity using a saturation binding assay, where a sub-nanomolar KD was reestablished. In vivo, 89 zirconium DFO RW03 was tested for tumor uptake by injecting it into HT29 tumor-bearing mice and counting organs for radioactivity after 24, 48, and 96 hours. The probe accumulated well in the tumor, reaching 15% ID per G, or injected dose per gram of tissue, after 96 hours, and showed excellent tumor-to-blood ratio, especially at longer time points. You can see characteristic blood and liver clearance over the study duration and low uptake in background tissue. With these results, we felt compelled, after a year of development and many challenges along the way, to test this probe for its capacity to image cancer stem cells. These are PET images rendered into animations looking for CD133 positive tumors in mice after 1 week post-injection for the first time in the reported literature. These three-dimensional volume-rendered animations show what it looks like when a mouse with a CD133 positive tumor is injected with our novel probe. We see liver uptake in line with the biodistribution study, and we see some uptake in non-target organs, specifically the bone, which our biodistribution study overlooked. But most importantly, you can see a large spheroid shape near the mouse's behind, which is the hind limb xenograft HT29 tumor. And you can see excellent tumor to non-target contrast, especially with non-target tissue in the tumor region. On closer inspection, the transverse view only identifies the tumor when the appropriate slices are taken, shown by the dotted line in the coronal images. The coronal view shows tumor uptake over 168 hours, with liver uptake decreasing owing to rapid blood clearance. In no images is the heart particularly visible. Over the course of 1 week, the probe increasingly accumulated in target tissues, reaching the highest percent ID per cc out of all tissues. I would also like to note that the y-axis spans 0 to 35% ID per cc, so all the raw data is shown without lower threshold filtering. While the entire tumor is visualized, even at this altitude, we can see that some parts of the tumor are more intensely visualized than others. Looking closer, our probe accumulated heterogeneously within the tumor. Images zoomed on the tumor show pockets of intense signal localized to the tumor core. We were pleasantly surprised to see this imaging pattern reflected ex vivo by autoradiography and by CD133 immunohistochemical staining. Physicians are increasingly concerned with intratumoral heterogeneity in their patients' lesions. Unguided biopsy samples risk mischaracterizing tumors. If biopsy samples from these HT29 tumors, for example, were taken anywhere other than the signal-intense regions indicated in red, might underinform physicians and mislead treatment. In addition to their usefulness in diagnostic imaging and companion diagnostics, These results illustrate how CD133 targeted probes may be used in combination with emerging high sensitivity imaging modalities like positron emission mammography to ensure physicians collect the most important tumor regions for biopsy. In conclusion, today I presented a synthesis and evaluation of a fully human antibody-based PET imaging probe for the rare and highly informative cancer stem cell population. Our probe was designed for clinical translation. It serves as a companion diagnostic for CD133 targeted therapy, where it's designed to stratify patients most amenable to CD133 targeted treatment. The antibody is also envisioned for theranostic applications, as its biodistribution and PET images closely reflect the biodistribution of lutetium DOTA-RW03, a targeted radionuclide therapy I'm also developing. Furthermore, our probe is designed for clinical translation and requires highly sensitive imaging modalities that we expect to be realized best with positron emission mammography. Before I finish, I'd like to thank everybody in McMaster's radiochemistry group for their help and support. I'd also like to warmly thank our collaborators, Dr. Sheila Singh, Dr. Jason Moffett, and Dr. Aaron Fenster. I'd like to acknowledge once more our commercial partners, the CPDC and Fusion Pharmaceuticals, with special thanks to STAR Imaging, who are associated with UHN. And finally, I'd like to thank the agencies responsible for funding this work, including the OICR, CIHR, and NSERC. With that, I'd like to thank everybody for their time.